Good afternoon, everybody. Welcome to a wonderful Wednesday afternoon of Chem 170 with your host, me, Dr. White. All right, game plan for today. First of all, I'm going to talk about upcoming attractions. Then I'm going to go over the amino acid protein chapter again. Then I'm going to take you on a special trip to a chemical plant, which I'm going to cut out of the video. And after that, we'll do the very last lab called carbohydrates. It really should be called starches, but anyways. And then after that, I will say goodbye and send you the password for test number 4. And you'll take it and also upload it by 11 o'clock a.m. tomorrow. And a couple other things I'd like to talk about. Next week, I'd highly recommend show up, because what I'm going to do, and it's helped students immensely, is I will do a review for the final, meaning the whole semester I'll review in 1 week. I can do that. I'm that good, maybe. But anyways, I'll do that by, first of all, Sunday, 3 p.m., if not a lot earlier, I will have test number 4 test scores posted in D2L, and also you'll get your individual email with your points for each answer on test number 4. By Sunday, the same time, if not earlier, I will post the final exam, not important, test, not final exam, exam information, how many points total, how many pages, how many questions, and then also I'll break it down, the points, 2 different ways. One will be like you've seen all semester, which would be general knowledge, like there's 4 bonds to carbon, those things you learn now. And there'll be nomenclature and reactions. And there'll be about 5 or 6 synthesis problems under reaction. I will also break it down by test material. How many points from test 1, 2, 3, 4, and then from new stuff, amino acids and proteins. Now, to make your life just a little easier, the final is set up that the first part of the final is from test number 1 material, second part, test number 2, third part, can you guess? Yes, test number 3, next part, test number 4, and at the end, new material, amino acids and proteins. And that's my test. Now, historically, when we do it face-to-face, even though I give students 2 hours and I'll give you 3 or 4, as long as you don't cheat, please don't. I know if you do, because I have secret things in there to tell me. And they work. But anyways, most students take about an hour and 20, hour and 15, hour, 20 minutes to finish my final. I'll talk more about it once I post on Monday the points and information about the final exam. It is a week from this Monday, and we will not have class. What I will do is I'll log in for a couple minutes just to say hello. If anybody wants to stop by and say hello, you don't have to. And at 1 o'clock on a week from this Monday, I will send out the password for the final exam. You'll have until Tuesday morning, 11 a.m. to finish it and upload it. All right, let's get going. First of all, any questions? Yep, I got my speakers on. When I first started in Zoom, I'd forget to turn them. I got these 2 huge speakers above here. They're big and amplified, which is why I don't have my hearing aids in. We were face to face, you'd see me with my bionic ears in. Speaking of that, on a serious note, if you know anybody who needs hearing aids, get them at Costco. The audiologist I was going to, because I had learned about her from my sister, to get new hearing aids, which I needed, would have been about $5,000 to $7,000. I went to Costco. It cost me $1,800. That's still a big thing, but that's almost 3,000 to 4,000 less. That's a real big thing. One advice, if you know anybody going to Costco, make sure you get the audiologist they have. Most stores have them. The one in Schaumburg, she's really good. Kathleen, there's a Kathy who's a technician, but get Kathleen, who's the audiologist, and she's really good. All right, what I like to do first is instant replay. And here I talked about amino acids, peptides, and proteins. Now here I have an amino acid, and another way of drawing it, I'm just swapping where the R group is in the NH2, the amino group. And this is the structure of all amino acids. R can be H or other things. And if we look at that carbon bonded to the carbonyl carbon of the carboxylic acid, the alpha carbon, that's always where the amino group is. It's amazing what Mother Nature came up with. And if you look at this, this has an amine group in it and a carboxylic acid. Now, when R is not hydrogen, which is glycine, when the alpha carbon, this one, when R is not hydrogen, even says it on the screen, then you'd have 4 different groups or 4 different atoms or combination, R, H, amino, carboxylic acid, that makes this carbon chiral. And most amino acids are chiral. They exist in 2 forms, like your right and left hand. Oh, it even says it on, and there can be an antimeric, mirror images. And you should know that. Now, I'm going to skip because it won't be on the final. Oh, I gave it away. But how do you bond amino acids together? By an amide reaction. And here, if we look at this part, we have a carboxylic acid. I'll call this double prime. This whole thing here is our double prime. And here we have an amine. And I'll call this whole thing here on this one R prime. And one of the hydrogens, I better make this triple prime because I'm using double prime over here. Uh-oh, I'm getting prime overdose. Look out. But anyways, one of the hydrogens, let's call it R double prime. And when you learn for test number 3, carboxylic acid, amine, you form an amide. Which is what holds amino acids together. It's an amide bond. Now you have, if we look at this molecule here, I can get fancy on you. I haven't done this all semester like this, but I will now. Here we have an amino group. Here we have a carboxylic acid. This can react with more amino acids on the red carboxylic acid. This can react with more amines right here. So you can build it out and out and out like a polymer. And actually, that's what you get, a polymer made of amino acids. Now we have names for those. The bond that holds between here, the amino acid of one, and the amino acid of another, protein chemists call it a peptide. I call it an amide bond because I'm not a protein chemist. And I'm never going to ask a peptide 2 or more amino acids held together. A protein is when you have 50 or more amino acids connected by, I call it amide bonds, you call it peptides if you're a protein chemist. Now, think about that. If I had amino acids, and I'm just going to use the letter A, get back to black. And you have 50 of them bond together. What's the key bond or functional group in a protein? And the answer is an amide. So next time you look at a steak, you're looking at a polyamide. Doesn't make it sound too tasty, but it is. Next time you look at your skin, you've got a polymer of amino acids, which are held together. Everybody else calls them peptide bonds, but they're really amides. Amides are holding your skin together or making your skin. That's scary. Now, one of the things, as you know, I like students to know is the real world, because we live in it, and organic chemistry is everywhere. And here you have what happens when you digest a protein. Let's just say if you're, what's the proper term for someone who's not a vegan but eats meat? Meatin? Never mind. But anyways, if you like meat and you've had a steak, hot dog, hamburger, or whatever, what happens in your stomach after you chew it and swallow it? Or if you're vegan, what happens if you eat some tofu, which comes from soybeans, which is mainly protein? Well, what's the key functional group in a protein? And the answer is an amide. Now I have it drawn here how my software likes. Here's an easier way to think of it. And you learn this for test number 3. And here, take an amide, react it with hydrochloric acid, which is actually the acid in your stomach, and water. If you've been drinking your water today, I hope you have. Speaking of that, I'm going to take a tea break. If you're not familiar, I get my tea from the Republic of Tea. This is a blueberry, wild blueberry tea. It's good. Anyways, you get back the carboxylic acid plus the amine salt of the amine you would have used to make that amide. Well, in the amino acid, they're connected together. And as you see on this slide, and I'm just showing 2, the same thing would be true if you had 50, 60, or 100. I have this amino acid or this peptide or protein, and here's my amide, HCl and water. This is going to be at the end. That's amine, it's a base, you get the amine salt, but this side becomes the carboxylic acid. This nitrogen becomes this amine salt, and what was attached to it, this is still attached to it. So when you digest a protein, what your body breaks them down to is amino acids. Now, I'm an organic chemist, and I haven't taken biochemistry or biology. Well, biology last time was freshman year of high school. That was a long time ago. So what your body does after that, exactly, I don't know, but it uses those pieces you get from your food to make protein, your skin, your muscles, all sorts of things. I like that, like Popeye. All right, leaving my class, there's something important you should know, and that is the following. Describe with words Two days later, I'll be finished with this. All right, you should know how to describe with words and a general reaction what happens when you eat protein. And what's in your stomach? Acid. You have water, so we'll call that hydrolysis. So we have acid hydrolysis. And what's the key functional group in a protein that gets broken down in your stomach? And that is an amide. And how do you describe this with words and a general reaction? Well, you learn for test number 3. Take an amide, react it with acid and water, then what do you get? The carboxylic acid plus the amine salt of the amine that you would have used to make that amide. Speaking about things on amine salts and carboxylic acids, I think I'll give till tomorrow, 11 a.m. I think it's on there, but I'm not sure, but I will. That's the deadline for you to get in your extra credit project if you plan on doing that. It's optional, but it's 10 points. Well, let's do that again. It's 10, hold on, 10 points. I had to sneak that in. I did. All right. What describe with words and a general reaction what happens when you eat something with a fat or oil in it, or both. And how do you describe that with words? Well, in your stomach, there's acid and water, so it undergoes acid hydrolysis. And what's the key functional group in a fat and oil? And that's an ester. So it's acid hydrolysis of an ester, the general reaction, which you learned for test number 3. I could have put HCl there, but just any acid. You have a ester, and you get back the carboxylic acid you would have used to make that ester, plus the alcohol you would have used to make that ester. So when you eat a fat and oil, how do you describe that with words? Acid hydrolysis of an ester. And the general reaction is an ester, acid and water, and you get back the carboxylic acid plus the alcohol you would have used to make that ester. And finally, the last food group I'm going to talk about for this question are the carbohydrates. And how do you describe with words and a general reaction. What happens in your stomach when you consume, ooh, fancy word from Dr. White, consume some carbohydrates. And what's in your stomach? Acid, hydrochloric acid, but acid, and water. So this is an acid hydrolysis. They all are. And what's the key functional group that holds different molecules of glucose together and carbs, carbohydrates? And the answer is an acetal. And here, the general reaction you learned for test number 2. You take an acetal or ketal, reacted with acid and water, acid hydrolysis. You get back the aldehyde or ketone you would have used to make that acetal or ketal. Plus, you get back the alcohol you would have used to make that acetal or ketal. And for those who need the 2 there, can I make that any smaller? I don't know, because if I were writing this for myself, I wouldn't put 2 there because organic chemists don't balance equations. So let's go through this again. Leaving my class, you should know how to describe with words and a general reaction what happens when you eat a protein. First of all, well, in your stomach, there's acid and water, and we call that acid hydrolysis. And what's the functional group that holds amino acids together in a protein? It's an amide. So it's acid hydrolysis of an amide. And here's the general reaction: amide, HCl, water. HCl is an acid. You get back the carboxylic acid plus the amine salt, the amine you would have used to make that amide. Now, the other food group you should know are fats and oils. And if you eat something with a fat or oil in it, or fat and oil, you have what? Well, there's still acid and water in your stomach, so it's acid hydrolysis. What's the key functional group in a fat or oil? It's an ester. So it's acid hydrolysis of an ester. Here we have an ester, acid, water. You get back the carboxylic acid plus the alcohol you would have used to make that ester. And the final food group we're talking about today is carbohydrates. And there, excuse me, and there, again, in your stomach, you have acid and water. Ooh, tea drink, break time. Boy, just talking about breaking down food is thirsty work. But anyways, when you have a carbohydrate in your stomach, there's acid, water, so it's acid hydrolysis. What's the functional group that holds glucose together in carbohydrates? And that's an acetal. And you have acid hydrolysis of an acetal. And what's the general reaction? And that is acetal or ketal. This is how you learned it for test number 2, acid and water. You get back the aldehyde or ketone that you would have used to make that molecule, the acetal or ketal, plus the alcohol you would have used to make that acetal or ketal. I just thought about something. 6 months ago, if I had said, oh, look at that ketone or look at that alcohol, would you know what I'm talking about? Think about how much you've learned this semester, and you should be proud of yourself. I am. All right, any questions on this? Going once, twice. Ooh, I can do it in stereo. 3 times. All right. It's time for something special from Dr. White. A clap. All right, the last lab of the semester. Oh, that's sad. I'm just getting to know you people. But anyways, last lab deals with carbohydrates. Let me just close some things. I hope you all enjoyed the trip to a chemical plant. Very few Americans, in fact, none who don't work at a chemical plant ever see the inside of that. All right, let's look at today's lab. Today's lab deals with carbohydrates. Now, long ago, like in the late 1800s, and a lot of labs we've done similar tests. How do you find out if something's in this food? And what we're looking for is starch. How do you know bread has starch in it? How about potatoes? How about the steak? Does that have starch in it? Well, how did they find out? Well, they found out by a color reaction, and they use tincture of iodine. Tincture of iodine is a solution of iodine, potassium iodide, and iodine. And when you make that, it turns blue, really a dark blue, so dark blue, it looks even black in real life. You got to look at it hard to see, wow, that's really dark blue. From the reddish brown color it is when added to starch. So let's look at a couple things important. Turned out they found that iodine, which is a crystalline solid, will do the same thing. What does it do? Starch molecule is really helical, not straight. Let me do a bigger one. And inside you have glucose molecules linked together, which have hydroxyl groups on it, alcohols. And if you get the iodine in there, it interacts with the oxygen in the helix, and it has to be in the helix. And because of that, it absorbs light. So you see a dark blue. But iodine is nonpolar. Instead of having to mix a solid and powder it and it doesn't work well, wouldn't it be nice if you had a liquid? You could put a few drops on something, and if it changed color, there's starch there. Well, what do? Well, you take potassium iodide plus iodine I2, and it reacts to give this molecule, potassium iodide, and this is polar, which means it's water soluble, because you know like dissolves like. And that's why you use tincture of iodine, which is this and really is this. And the same thing happens. This is a red-brown color. Depending on concentration, it looks red, looks more brown, but reddish brown. And when this gets into a starch, it now turns a very dark blue. And today's lab, you're going to use tincture of iodide to find out if certain food groups have starch in them or not. Time out for
a family vacation in a verbal meeting.
one second.
The form of starch.
Everybody see the gentleman on the screen? Thumbs, thank you. All right, let's play it.
An important constituent of our daily food are carbohydrates, which we consume in the form of starch. Starch is a high-calorie food that converts to sugar during digestion. Starch is also produced by all green plants as an energy store. Today, we will conduct an experiment to test the presence of starch in potato, rice, sugar, and salt. For this, we will need iodine solution, which is yellow-brown in color.
I call that
Note the
reddish
change
brown.
in the color of iodine as it is put on the rice and potato. Both turn the iodine blue-black. Put some iodine on the sugar cubes. The color does not change. Salt too does not change its color. This proves that sugar and salt do not contain starch, while the starch in rice and potato reacts with iodine to form a blue-black compound. Now, the reaction is because the blue-black color is basically the formation of polyiodide chains from the reaction of starch and iodine.
Now, in today's lab, you're going to be taking on a crystallization just looks like a little plate, small amounts of the various food groups to be tested. And you add a few drops of tincture of iodine, wait 1 minute, and look at the color. Now, since you don't have that at home, here's the stuff you would have tested. Potato, bread. Now, I'm going to tell a story about difference between yellow and white cheese, flour, cereal, honey, sugar, honey, and cornstarch. And here's the data you would have seen if you were doing this real life. Now, a couple of funny stories. First of all, ECC, to buy it, it's the lab manager really has a hard time buying things like this. So some of the stuff he'd bring in, some of it I'd bring in. And funny story, for bread, I would bring in matzah because it lasts forever. Well, the first semester we did that, the jar that had the broken piece of matzah, the bottle, it's actually a jar, had the word matzah on it. And the students came up to me, Dr. White, where's the bread? I realized, oh my gosh, I'm the only Jewish person in the lab today who knows what matzah is. So the lab manager, Dr. Daly, who does a great job when we're face-to-face for my labs and everybody's labs, put on their bread and in brackets underneath matzah, and people knew what to use. All right, now, I would bring in cheese. In the past, I'd bring in a white cheese, and I would do that. Well, one time I had to get more cheese. So the local place I shop at, Valley Food Market, had a sale on the shredded yellow cheese. I don't know which one it was. So I bought some, and the students, Dr. White, why is the cheese turning blue? I said, well, let me watch you do it. You did something wrong. And it didn't, it turned blue. I went and looked at the package, and guess what was on the label? The ingredients contains starch fillers. So I also bought more expensive white cheese the next time, and they did both. And notice the white, more expensive, doesn't have any starch in it. All right, and for this lab, here are the questions you have to answer. And with that, I'm done today. Remember, if you need any help or anything, I will have my office hour tonight from 5 to 6:15. I will also check my email on a regular basis tonight and tomorrow morning before I go to teach at College of DuPage. by Sunday, 3 p.m., I will have your test, if not earlier. I'm shooting for a lot earlier, but I guarantee by then, even if I got to stay up all night, Sunday or Monday, Saturday night, I won't. But that helps me make sure I get things done. I will have your test scores in D2L. I'll have mailed out individual emails with your points for each answer on test 4. Now, this weekend, I highly recommend you start studying for the final exam. You don't want to just cram the night before. It's better to spread it out and practice. Now, if we were face to face, you would have been allowed, I would pass back and allow students, I always do, keep the hourly exams. And you've already got yours. I hope you didn't delete the file. And that hourly exams, 1, 2, 3, and when I give you back, you have 4, use those as a practice problems for the final exam. You can go back and use the regular practice problems. Am I going to use the same problems on the final? No. Am I going to use similar ones? Yep. I've done that all semester with the practice problems. So use your hourly exams as practice problems. And with that, I'm done. I'm going to say, Gang gesund. I'll see you on Monday. Next week on Monday, I will go through the answers to test 4. But as I've done all semester, I cut that out of the video. If you need help, come to my office hour or make an arrangement, see me at another time. Take about 2 hours or so to finish test number 4. And as soon as I shut up and log off, I'm going to go send out the email with the password so you can get started when you want to. With that, I'm done. Gang gesund. Good luck on the test. Hopefully you won't need luck. Bye.
Thank you.
You're welcome.